Started.
In 1 minute, I'm going to give a quick update. Okay, my name is Amber Freed. I am the Executive Director for Health Fountain Syndrome. This is the July virtual roundtable. We just did a quick round of introductions, and of course, Dr. King will announce his work before his presentation. Our big conference is coming up. We are so excited. It is September 24th, the day before Prader-Willi begins. So a lot of people will be able to just extend their trip one day. And of course, we can all stay through the entirety of PWS. So super excited. After I'm done giving updates, I will put the link to register into the chat. Bo is not able to join today. Hopefully he'll be able to sign in though. I want to point out that he gives a weekly podcast. He's never missed a week, and it's 10 minutes, very to the point, utilizes Bo's incredible storytelling ability. And it really walks you through what it's like living with a rare disease child, and you get a glimpse into sweet little Tessa's life. And I'm trying to highlight a patient every week just so that we're very connected to what daily life is like. And please meet Ella, and her mother, Becky, is signed on. Ella has a health update, and Becky was going to give us a little update. What's going on?
So this is Ella and our dog Napoleon in the photo. So Ella is probably definitely considered a higher functioning Howe Fountain patient. She's just been mainstreamed in school this past year. She's going to be going into 6th grade. However, she had a pretty bad seizure. I want to say it was in April, where the seizure was for an extended period of time, and then she completely lost her vision for about 5 minutes after the seizure. So her neurologist had her go in and have another MRI. Ella's had many, many MRIs because of the brain changes. And unfortunately, Ella has now been also diagnosed with mesial temporal sclerosis. So we were kind of, that news was kind of upsetting for us as parents to hear. And she also did have some additional changes in her brain where they're not technically abnormally sized, but they told us that the ventricles changed, which they're not quite sure if that means there's some white matter loss. They weren't sure. We'll go back and I'll talk to them again to see what that means for her. She's also had some weird blood things where we now have to give her carnitine supplements because she's now deficient in that. And she's got some iron deficiency anemia. So she's got a bunch of things kind of new to her that are going on. And really, in these updates for her, we're just living through it daily. But I wanted to also take this time to really thank everybody who's on this call, who is out here, doing this research, and you care about USP7 and you are caring about our kids. And it's so important for us and for her, to find out what's going on. Are there going to be other kids that are going to have some of these changes? We don't know. So, I just wanted to thank you so much for doing this work because it is really, really important. It's really important to our family. It's really important to Ella. And if you have any questions about her, I'm an open book about anything with her. I'm willing to share anything.
Becky, I was just curious, is the carnitine deficiency related to the anti-seizure medications, or do they know what's going on with that?
Um, so they didn't really say, but in my research, because she's on oxcarbazepine, it doesn't seem like that is typically one that would cause that. But when we go back for our next follow-up, she's going to go back again in just a couple months. I'm going to ask about that to see if it could be that, because I did read that myself in, of course, Google is every parent's friend and nightmare.
Yeah, I know valproic acid can do it, but I'm not sure all the different agents can do it.
Yeah.
Okay, we also have some funding opportunities coming up. There are travel grants available through Uplifting Athletes. You can also apply for grants through Uplifting Athletes. They actually have a full scientific draft in Philadelphia, and it's much like football, which I've never watched one, but I can tell you the scientific is super fun. Like so much energy, people from the NFL come, and everybody just celebrates all of the work being done. Then there are some grants through the American Epilepsy Society. And interestingly enough, the Centers for Disease Control has some funding opportunities right now, and I will send out links for all of these on the wrap-up email. Our goals for this year, we're working on further understanding the mechanism of disease and establish correlations with larger communities like cancer. We're conducting a small molecule screen. We did apply for an ICD-10 code, and we plan to publish a disease concept study. There was a new paper published out of Dr. Schaaf's lab, which was pretty interesting. I'm sure most of you saw this, but I will include the link in my follow-up email. And now it's time for Dr. King's presentation this morning. And I do not want to do you the disservice by introducing you myself. But I did get the pleasure of speaking with you and was just really blown away by how much you care and how much empathy, and that you're every rare disease parent's like best situation because of just how validating and kind you are, and how you can just feel how much you care about our kids. And that doesn't even touch upon your brilliant scientific and medical mind. So before you even start, just thank you. And I will stop sharing my screen.
Thank you, Amber. You're very kind. It was really a pleasure to talk to you a month or so ago. And in addition to running a research lab, I direct a course for first-year medical students that spans most of the basic science disciplines, runs 13 weeks, and is coming up here in the middle of August, right around the corner. And part of that course, we have a weekly patient clinic where we invite patients to come in and really explain their experience of disease, so that students really understand not just the science of it, but really how it impacts families. And many of those sessions indeed are rare diseases. And Amber's actually going to help wrap up our course this year and come and tell her story to our students. So grateful that that she's willing to do that. So let me share my slides here. So just to give you a little bit of background, you know, I'm really a cell biologist and a chemical biologist, and our goal is to use chemical tools to really understand the function of proteins, really prior to a point of I would say, a pharmaceutical company level validation of targets. So it's really chemical biology as a tool to understand biology. And as part of that work in the past, we've developed inhibitors of certain deubiquitylating enzymes, including USP14, which is an enzyme that sits on the proteasome and can remove ubiquitin chains from substrates prior to their degradation. That work we've done for a long time in collaboration with Dan Finley in our department. But as a result of some of that work and other studies that we've undertaken, we wanted to try to develop some approaches that would allow us to identify substrates of deubiquitylating enzymes in an unbiased approach. And so we've been working on this for the past 6 or 7 years or so. Most of this work has been spearheaded by Valentina Rossio, who's a postdoc in the lab. And we've had a long-standing collaboration with Steve Gigi in our department, who's really a terrific quantitative mass spectrometrist, and one of his trainees, Joe Apollo. So these are the folks who've really done the work that I'll tell you about today. So you all probably don't need this slide. I think you probably know at this point that deubiquitylases are enzymes that remove ubiquitin from other proteins, reversing the effects of E1 and E2 and E3 that conjugate ubiquitin onto proteins. And ubiquitin conjugation can play a large number of different roles in the cell. And this really depends on the protein and the type of ubiquitin attachment. In some cases, ubiquitin can be attached in the form of polyubiquitin chains, and certain chains can target proteins for degradation by the proteasome. In contrast, we're learning more about proteins whose activity is controlled by ubiquitylation. In this case, it might involve the addition of one or a few ubiquitin molecules. And it's known, probably the best example is the ubiquitylation of histones that can influence gene transcription. But one of our goals in this project is really to kind of expand our repertoire of proteins that are ubiquitylated that might control their activity rather than simply targeting them to degradation. So again, you probably all know very well that DUBs can work by different mechanisms. Some are highly ubiquitin chain specific and will remove ubiquitin from ubiquitin chains. In contrast, many DUBs seem to work more by recognizing protein substrates and removing ubiquitin from those substrates in a much less ubiquitin chain selective fashion. So again, our goal was really to develop a method that can identify new DUB substrates and also to directly compare the activity and specificity of one DUB to another. There's a lot of wonderful work on DUBs, including work from Tizia's lab, who's really been a leader in USP7 and understanding its mechanism and function. But in a lot of cases, DUBs are studied in isolation. Labs focus on a particular DUB and in a particular physiological system. And so it can be a little hard really to understand whether DUBs have a broad reach. And so that was kind of one of the questions we wanted to address in this study. So there are about 100 different known DUBs, and most of these are cysteine proteases, a subset are metalloproteases. For the purposes of this talk, it's important to know that the cysteine protease class can be inhibited by a covalent inhibitor called ubiquitin vinyl sulfone. So it's quite selective for deubiquitylases, but the advantage for us is that it can broadly inhibit classes of deubiquitylating enzymes. As I mentioned earlier, some DUBs are non-chain selective, and most of the USP DUBs, which is the largest category of DUBs, are non-chain selective, although there are some exceptions. In contrast, other classes like the O2 class are highly ubiquitin chain selective. So in the work I'll tell you about, we profiled 30 different dubs, basically all the dubs we could get our hands on as purified recombinant enzymes. And you can see it represents most of the classes of dubs. We had, for technical reasons, difficulty screening the UCH dubs. So that's why we didn't screen any of these. We didn't screen the metalloproteases because they're not sensitive to ubiquitin vinyl sulfone. So this is a summary of the number of known substrates, a very nice review from the Miori lab back in 2022. This is a nice resource for anyone who's interested. And so what you can see here is that some dubs, like USP7, have a lot of known substrates, and that's in part because it's a well-studied dub. In contrast, other dubs have relatively few known substrates. And again, one of the questions we wanted to get at is, does this heterogeneity really, does it reflect the intrinsic activity of the enzyme, or is it just that we've really studied some of these enzymes a lot, and that's the reason why there aren't many, there are more published substrates for those enzymes relative to others. So I'll come back to this point in a bit. So our approach has been to use the Xenopus egg extract system for these studies. And what we do is we isolate eggs from Xenopus laevis. These are large eggs, about 1 millimeter in size. And we crush them in a centrifuge to obtain this middle layer, which is essentially close to undiluted cytoplasm. And the reason we use this system is that there's no new transcription or translation in this system. There are no nuclei that are present. And so the idea is if we perturb the system biochemically, the effects that we see are more likely to be direct biochemical effects rather than effects that are mediated indirectly through transcription and translation. The other reason this system is valuable is we're inhibiting new protein synthesis with cyclohexamide. And so we avoid complications from the impact of dubs on quality control pathways. So we know ubiquitination is really important in targeting misfolded proteins for degradation when they're getting synthesized. And so it's our feeling that this is kind of a simplified system that allows us to look more directly at the biochemical effects of dub addition. And I should say, this system has been used in the past extensively in cell biology to reconstitute very complex processes. In my graduate work and work in our lab, we've used this system to study regulation of mitosis and E3 enzymes like the anaphase promoting complex that are important for driving mitotic progression. So we've had a lot of experience in these using these extracts for other approaches. So the basic idea here is to first we treat these extracts with this broad spectrum dub inhibitor ubiquitin vinyl sulfone. And so the idea here is to try to lower endogenous dub activity in the extracts. And here what we're really trying to do is to avoid complications of functional redundancy of enzymes. It's known that at least certain dubs seem to have overlapping sets of substrates. And because of that, if you take genetic approaches to knocking out single dubs in cells, because other dubs may be able to basically step in and target the same substrates, genetic approaches have the potential to miss particular substrates. So the idea here is to kind of broadly inhibit dub activity. And then what we do is we add back individual dubs. And so the idea is here, we're really testing sufficiency. What proteins can an individual dub target in a background where we've lowered overall dub activity? And to find those proteins, when we add the recombinant dub back, we add a tagged form of ubiquitin, HA-tagged ubiquitin, which will get incorporated into endogenous ubiquitin protein conjugates. And so we isolate those proteins using an anti-HA antibody. We elute them with an HA peptide. And we do all this under very stringent wash conditions, although it's not a denaturing purification. So we're able to isolate protein complexes, for example, under these conditions. And then we quantify changes. We basically compare the effect of adding the dub to no dub addition, and we quantitate differences by tandem mass tag mass spectrometry, an excellent quantitative method. And so a typical experiment looks like this, where we'll test the effect of no dub as a control to the effects of adding one of three different dubs. And we do these experiments in quadruplicate, so we can get statistical estimates of variability in our system. And then a candidate dub substrate would look something like this. If we had a dub 3 in this case working on a substrate, the idea is it would have less ubiquitin attached, and as a result, we would isolate less of the protein when we do our pull-down experiment. So overall, we did 10 experiments with 3 dubs each for a total of 30 dubs. For a subset of the dubs, we also went in and repeated the experiment so that we would have independent entire repeats of the experiment. I'll show you a bit of that data as well. And feel free to please interrupt if you have any questions about anything as I go through. Okay, so that's the overview of the method. So I have some slides just focusing on USP7 here, given the audience. I thought it would be a good one to focus on. So each experiment for each dub, we can generate a volcano plot, which just shows the log 2-fold change comparing USP7 addition to no dub addition. And so if proteins are reduced in abundance, they'll be shifted here to the left on the plot. And this shows the statistical significance of our measurement. So we want things above this dotted line. So the proteins that are changing in response to USP7 addition to these extracts would be shown here in the upper left quadrant. Now, to be able to compare dubs across different experiments, we calculated a percent impact, which is the number of candidate substrates. These are the proteins that are reduced by a significant amount, divided by the total number of proteins that are identified in the experiment. So in the case of USP7, the percent impact was 18.4%. That was actually the highest impact dub that we found in the screen. So if we now look across all the experiments for all the dubs, this shows the percent impact for each dub. We've color-coded the dubs by class here. And you can see we find 5 dubs that fall into this high impact category, a number that are medium impact, and most are lower impact. Right away, you can kind of see how most of the dubs on the top are blue, or they're of the USP class. So again, this is consistent with the idea that it's thought that most of these dubs are non-ubiquitin chain selective. In contrast, you can see the O2, the MJD classes were typically lower impact dubs, with the exception of ZRAN-B1. And again, most of these dubs are thought to be ubiquitin chain selective. So we think that most of the high and medium impact dubs are likely working by recognizing features of the substrates, the substrate proteins, rather than any specific aspects of the nature of the ubiquitin itself or ubiquitin chains that might be attached. Okay, so now what I want to do is just go back to this diagram I showed you earlier. Again, this is the number of published substrates in the literature. And what I'm plotting here is the percent impact that I showed you on the previous slide. So this essentially allows us to kind of compare our results to the literature more broadly. So again, as I mentioned, we found USP7 as the highest impact dub in the screen, perhaps not surprising given the number of known substrates of this enzyme. Similarly, we found a lot of candidate substrates for USP9, for which there are a lot of known substrates. However, we found several dubs, USP15, USP36, USP24, that also had a high impact, but have a much smaller number of known substrates. So we think that these enzymes, perhaps because they've been less studied, may have fewer reported substrates. Or there could be other features that constrain their activity in, for example, human cell systems. And I'll come back to that point a little bit later. You can see here an interesting difference was USP14. This is an enzyme we've studied a lot. And you can see we found very few USP14 substrates relative to what's reported in the literature. And this actually makes sense because we think that, I should say, we know that USP14's main role is to sit on the proteasome and regulate the degradation of proteasomal substrates. In our system, we think we're isolating mostly non-proteolytic substrates that are being modified by ubiquitin, but that aren't being targeted for degradation for the most part. And so I think this lack of effective USP14 is actually consistent with the known function of USP14. Okay, so as I mentioned, for a subset of the dubs, including USP7, we did an entirely separate independent experiment and then looked at the proteins that we identified as candidate substrates in 2 independent experiments. So in that case, we found a total of 116 different proteins. And those proteins fall into a variety of different functional categories. Many of them are E3 ubiquitin ligases. Much of this work was done by Titia to kind of define a sort of important relationship between USP7 and a variety of different E3s. But you can see there are a variety of proteins that have other functions that we also identified as substrates. Now, shown in green here is the number of USP7 known substrates in the literature that overlap with the set that we identified. So everything in green here is a protein that's been reported in the literature previously as a USP7 substrate. So overall, it's 46 out of 116, which is 40%. So a pretty large degree of overlap, I would say, given certainly the differences in experimental approaches and physiological systems. But we think this is nice evidence that the Xenopus system can actually identify bona fide USP7 substrates, and that the features of substrate recognition may be conserved across different experimental systems. Okay, so at the end, I can take a moment to show you a web-based resource that we've developed to let people access this information. But one of the plots you can perform using this web resource is to look at the effect of a given dub. So here are the 30 different dubs across different substrates. So here are 3 different proteins, and these are the human orthologs that I'll show you in just a second, and what their profiles look like in the Xenopus experiment. So here's HSM-NDC1, which showed up as really quite a selective USP7 substrate. HMGB2, again, fairly selective for USP7. And then WP2, which was acted upon by USP7, but also USP24. So if you have a protein you're interested in, you can just look it up and sort of see what its profile looks like. So one approach we've taken to validating substrates is to express the human orthologs in rabbit reticulocyte lysate, where we can radiolabel these proteins, run them out on a gel. And this is what it looks like if we incubate these proteins in the extracts where we broadly inhibit dubs and add ubiquitin back to rescue ubiquitin levels. You can see these higher bands that represent ubiquitination of these proteins. And then if we now repeat that experiment under our screening conditions where we add USP7, or in this case, USP21, we can see a reduction in the ubiquitination of each of these proteins. Whereas USP21, in comparison, has relatively little effect on these 3 different proteins. So in any case, this is one approach we can take to basically validate the results in the mass spec proteomics screen. But a nice feature of this is it then gives us a tool to go in subsequently, using mutagenesis of these substrates, try to identify regions that may be ubiquitilated, and also to identify motifs in these proteins that might be responsible for their targeting, either to ubiquitination or to deubiquitination. Okay, so I think that's all I'm going to say specifically about USP7, but I'd be happy to answer more questions about that at the end. But I just briefly wanted to give you a sense of the overall data set and what it looks like. So as I mentioned, we did 10 different experiments, and this shows the number of proteins that we isolated in each of the experiments. And you can see here there was a set of about 1,000 proteins that we isolated in common in every experiment. So we think this is a set of proteins that gets ubiquitilated very reproducibly under our conditions. And we thought this was a subset of proteins that would give us a nice opportunity to really compare activity of DUBs across the whole screen, because these are proteins that we found in every experiment. So we call this the common set of ubiquitilated proteins. So if we now look at this set of 968 proteins, what we found is that 374 of them were substrates of at least one DUB. In contrast, 594 were insensitive to all the DUBs that we tested. So this is kind of interesting. I'm going to we talked earlier about how USP7 was actually quite an active DUB. Yet there are still, 60% of the proteins that we isolate are insensitive to everything we test. So there are interesting levels of specificity that are operative in this system. If we look at the 374 proteins, we can then ask how many DUBs acted on those proteins. As I mentioned earlier, it's known that some DUBs can function redundantly against certain substrates. So we wanted to get a sense of what is the overall level of redundant action in the system. So of these 374 proteins, 124 were acted upon by only one DUB. Okay, so at least for a substantial fraction of the proteins, there seemed to be a fairly high degree of selectivity. However, there were a substantial number of proteins that could be acted upon by 2, 3, 4, or even more DUBs, with some substrates that seem to be targeted by many of the DUBs that we screened, showing highly redundant effects. And I'll talk a little bit about some features that may explain that behavior a little bit later on. All right, so one thing we did is we took this common set of proteins and we said, well, what kinds of proteins are present in the common set? So these are functional categories of different classes of proteins, similar to the classes that I showed you earlier with respect to the different classes of USP candidate substrates that we identified. And so you can see here in gray is the number of proteins in each category. So these in gray represents the proteins that we're reproducibly isolating across all these experiments. And in green, we're showing the number of those proteins that are candidate substrates. Okay, so a large green bar would mean we're isolating a large number of candidate substrates in that class. And then what we do is we simply divide the number of candidate substrates by the number of proteins in the category. So we divide the value of the green bar by the value of the gray bar to calculate a percentage. And what that does is it tells us the percentage of proteins in that category that are sensitive to DUBs. And we've now arranged these categories by that sensitivity. Okay, so the proteins categories at the top tend to be highly DUB sensitive, whereas the categories at the bottom tend to be relatively insensitive to DUBs. So if you look at this, it's kind of interesting. Again, as I mentioned before, USP7 has a number of proteins that are E3 enzymes as substrates. And you can see that in general, E3s were quite sensitive to DUB activity across our screen. 57% of the E3s were substrates of at least a single DUB in our screen. In contrast, if you go down here to metabolic enzymes, This is quite interesting because we identified a lot of these proteins, over 100, reproducibly across these experiments, yet only a small fraction, 7% of them, wound up being substrates of a given dub. So there's something about metabolic enzymes that renders them relatively resistant to dub activity relative to proteins higher up on this list. Okay, so then first thing we can do now is, again, look at those protein categories. These are the same categories that I just showed you. And in color is the sensitivity of the category to all or any dub. So this is basically, in this first column, just a recapitulation of the data that I just showed you on the previous slide. But now what I'm doing is breaking it down by each dub. Okay, so this shows the sensitivity of each of these categories to each of the different dubs that we've tested here. Okay, so you can see, for example, for these centrosome centriolar proteins that are overall quite sensitive to dubs, you can see they're highly sensitive to USP9, to USP36, a little less so to USP7, and not very sensitive to USP15. We can look at other classes of proteins like ribosomal proteins or the proteasome. And we found that ribosomal proteins are sensitive really to a very discrete set of dubs, and the same was true of the proteasome. So I'll dive into a few of these categories in a little bit more detail, but again, I'm going to try and stick, for the purposes of today's talk, at sort of a higher level of abstraction. Okay, so again, this is really how different categories of proteins are sensitive to different dubs. What this represents is hierarchical clustering of the data set for the 968 common proteins. So on the vertical axis here are the 968, sorry, it's not the 968, it's the 374 proteins. It's a subset of those. So to be in this chart, they have to be a substrate of at least one dub. Okay, so what we're saying is, amongst all the candidate substrates, how are they acted upon by these different dubs? And again, we've done hierarchical clustering, which basically puts dubs with similar patterns together across the horizontal axis, and substrates that have similar behaviors on the vertical axis here. And again, the candidate substrates are shown in gray, and purple would mean that the protein was not a substrate of a given dub. So I just want to show a few general features of what we found here. Okay, so I mentioned earlier this set of high impact dubs, right? These are dubs that act on a large number of substrates. Those dubs all clustered together in this part of the clustering here. And that's because they both had a large number of substrates, but they also had a large number of substrates shared in common, although they didn't share all substrates in common. Okay, so we think these are substrates that different high impact dubs can all access. However, each of these high impact dubs also showed unique substrates. So here you can see proteins that were specific for USP15. Here's a set that were specific for USP7. Here's USP36 specific, USP24 specific, and USP9 specific. Okay, so again, we think these high impact dubs may be recognizing a common pool of substrates through some shared feature, but they also have the capacity to recognize an independent set of substrates, perhaps mediated by specific protein-protein interactions between the dubs and those proteins. Other categories that we found here, USP8, 10, and 16, turn out to be quite selective for small ribosomal subunit proteins. And in fact, USP10 and USP16 are known ribosomal deubiquitolases, but a function for USP8 in targeting the ribosome has not been established. These dubs were medium impact dubs, so you can see they have fewer substrates overall. There's less gray in here than there was for the high impact dubs, but some of these in fact overlap with the high impact dubs. And then here we have the low impact dubs, and most of these proteins tend not to overlap with the high impact dubs, although a few do. And then finally, I just wanted to mention the proteasome. These are the proteasome subunits here. These proteasomal subunits seem to be targeted by 3 different dubs in different categories in the screen. So that was kind of interesting as well. All right, and I just want to wrap up with a few comments about what is it that it might explain the ability of these high impact dubs to recognize many shared substrates? And as I mentioned earlier, why are some protein classes like the metabolic enzymes just insensitive to dub activity? So to address this question, we looked across all of our substrates and looked at different features of those substrates and asked, do the substrates share any specific features in common? And the one that stood out to us was the percentage of disorder. Okay, so what this means is proteins can fold into a very well-folded domains, and those domains can have a variety of different structures. But it's clear that proteins also have disordered regions that don't fold into stable 3-dimensional folded domains. And those regions can be very important as sites of post-translational modification or protein-protein interaction sites. And so what we found is if you compare the non-substrates to the substrates, that in general, the substrates have a higher level of overall disorder. They tend to be more disordered. Furthermore, if you ask and compare substrates that are substrates of 1 or 2 dubs versus 3 to 5 dubs versus 6 to 13 dubs, so these are the proteins that are acted upon by many dubs, you tend to see an increase in disorder that correlates with an increase in functional redundancy. Consistent with that is we compared all the substrates that were sensitive to the high impact dubs to those that were insensitive to the high impact dubs. So in this case, these are substrates, but they're recognized by non-high impact dubs, either the medium or low impact dubs. And again, you can see that sensitivity to the high impact dubs was correlated with an increase in disorder as well. And then finally, if you look across those different functional categories that I mentioned earlier, some of the categories like the centrosomal HNRMP proteins were highly sensitive to dubs, meaning all the proteins that we identified turned up as dub substrates. You can see that these classes in general are highly disordered. In contrast, categories that were insensitive, like the metabolic enzymes, most of these proteins are not substrates. A few are, but most are not. You can see again that most of these proteins tend not to be very disordered. And then we thought it'd be interesting to go back to this literature data set and say, well, would you find the same thing if you looked at the known substrates, the published substrates of USP7 or USP9, which we identified as high impact dubs in our screen? And again, so this is the calculated percent disorder of published USP7 substrates. And over here is our data for the USP7 and USP9 substrates that we found in our screen. And you can see here that there's basically almost no difference across these different protein categories. They're all relatively disordered. And in contrast, we thought it'd be interesting, okay, well, let's go back and look at USP14. There's a lot of USP14 substrates, but as I told you earlier, most of those USP14 substrates are targeted at the proteasome for degradation. And in fact, those tend to be lower disordered proteins. So we think this is, in fact, a feature of some of these substrates, maybe a sort of a shared feature, not only of what we're finding in the Xenipa system, but potentially of other published substrates as well. So the last point I'll make before wrapping up here is just to raises a very important question, like how are these high impact dubs doing this? Like how do they recognize all these substrates? And we don't have a good answer for that yet. But one thing I'll point out is that if you just look at a simple parameter, the size, the length of these dubs, there's a pretty good correlation, in our value of 0.627 here, between dub size and impact. So USP9, USP24 are very, very large dubs. They were both high impact dubs. USP7, 36 and 15 are a little smaller and were also high impact here. But they're larger than many of the other dubs that we found in our screen. So this could be because the high impact dubs have more protein-protein interaction domains that may be able to facilitate recognition by the substrates. So then just to summarize everything here, we found a set of these high impact dubs, including USP7, that seem to be able to recognize a pool of shared substrates, but these enzymes also seem to have independent substrates that they can recognize in a selective fashion. We've identified dubs that can recognize protein complexes like the proteasome or the ribosome. For the highly chain-specific dubs, we didn't identify many new substrates for these enzymes, but we identified some, and those seem to be largely non-overlapping with the other sets of substrates that we identified. And then finally, we have a pretty large set of ubiquitinated proteins that seem to be insensitive to the dubs that we've tested. It may be that we just haven't tested the right dubs yet, so we're kind of continuing with this screen. There are many dubs still to go, but it also may be true that there are features of these proteins that make them less sensitive to dub activity. And sort of understanding how the degree of protein folding of these proteins influences their sensitivity, I think, will be an interesting mechanistic question to pursue in the future. So I'll stop there and just refer you to a couple of papers. This paper we published very recently. This other paper we published a number of years ago that looks more broadly at ubiquitin dynamics in the Xenopus system. And if anybody is interested in more details about any of these proteins, I would refer you to this website that we set up that allows you to really access all the data and profile these dubs if you're interested in. So I just want to thank some of our colleagues, Martin Wurr and Mark Kirschner, who really laid the groundwork for the Xenopus proteomics that we built upon. And then funding from NIGMS and the Goldberg Fund here at HMS. So I'll stop there and happy to answer any questions.
So what is the next step?
Well, I think for us, the two things that we're interested in doing are, continuing the screen. We're now at a point where we've sort of exhausted commercial sources of these enzymes, so we have to start making them ourselves. But the good side of that is we can now also start to do structure function analysis to make mutations in dubs, and ask how it influences the spectrum of activity that we see and specificity that we see. So that's what's on our agenda, but certainly welcome thoughts or ideas. Dithya?
Yeah, it's very interesting. And I think it will be very useful, even more so if you get more dubs in, I guess.
Yeah, right.
The question I had, you say, okay, you have these some proteins that had this disorder, etc. No, yeah, also is the difference in these proteins that get ubiquitinated in the targets, is it in the ON process or in the really in the dubs?
Well, so when we do that enrichment for disorder, when we do that analysis, that is within the set of ubiquitinated proteins that we identify.
Precisely,
okay,
but that could also make them better targets for the E3 ligases for the E2.
It could, but again, we're seeing this enrichment comparing the substrates to the non-substrates. And these, again, this is the set of proteins that we're reproducibly identifying. So
Yeah.
this is within the population of ubiquitinated proteins. We've tried to do the comparison you're talking about, which is to do if you compare the ubiquitinated proteins to the Xenopus proteome, are those proteins also more likely to be disordered? There's some enrichment there, but it's not as strong. It's a little bit of an enrichment, but we're isolating a lot of proteins that don't have a high degree of disorder as well. So I think the enrichment, I think you're right. I think it makes sense that at least some subpopulation of ubiquitinated substrates are more likely to be disordered. That sort of makes inherent sense to me. But really what the data are telling us is there's also a substantial number of proteins that get ubiquitinated under these conditions that are not very disordered. And that among those proteins, among the total set of ubiquitinated proteins, there is a further enrichment if you look at the DUB substrates relative to the non-DUB substrates. But again, I think a lot of that enrichment is coming from this subset of substrates that are being affected by these high impact DUBs. So, I think disorder could be playing a role at multiple levels here, as you're asking, but I do think there is something about the DUBs that is enhancing their ability, at least a subset of the DUBs, enhancing their ability to be able to access and deubiquitinate some of these proteins.
Yeah. And do you think the categories that you now find that are poorly deubiquitinated by this set of 10 DUBs have their own selective DUBs more likely?
I think that's a great hypothesis, and I think we're particularly interested in figuring out what's going on with these metabolic enzymes.
Exactly. Yeah.
Again, I just don't know. We've done, I'd appreciate any thoughts you have about this, but I don't know. I think it'd be interesting to go through, and we just haven't had time to do this systematically, but really go through, well-characterized DUB substrates that are metabolic enzymes, and for published examples, have people been able to define features of those proteins that allow them to be recognized? So we're not sure. I will say there's a caveat here, that this, like any technique, it's not perfect. And if what our screen would miss are enzymes that partially deubiquitinate substrates, right? Because we just have to have maybe 1 or 2 ubiquitins on a protein to be able to pull it down, right? So it's possible some of these DUBs are going in and removing a few ubiquitins, maybe in a specific way, but maybe aren't able to completely deubiquitinate the substrates. So they're not reduced very much when we look at them under our method. So that could be a piece of it. But I think we'll just have to, do some more work to try to figure that out.
And so going back to USP7, so 2 questions I have is how much is or 1 question, but there are 2 possible levels. How much is this protein regulated? When is it on and when is it off, right? And because it has that very special activation through its C-terminal tail, my feeling is always you need to bring that in, and there may be moments it's not. Still, there's an enormous number of targets, as you show. So it's often on, but I could imagine there are conditions where it's not. I think that for the tail, I don't see an immediate answer for you, but we've also collaborated once with a group that looks at phosphorylation of USP7, and that you could test, I guess. Are you planning to do that?
Well, we don't know what the post-translational modification state of USP7 is in the extracts, but I think if we had a tagged protein, we could assess that. It would be straightforward, I think, to add USP7 to the extracts and re-isolate it and ask it if it's put post-translational modification state is changing.
Yeah.
You know, in terms of the activation, it's an interesting question. You know, there may be endogenous activators present in the extract. I mean, it came out as the most active dub in our screen, so I think it's working really well. So I wouldn't be surprised if that's true. But you know, I think
I think one thing. Sorry to
No, go ahead.
interrupt. USP7, I think the levels matter enormously, and that is a question. I haven't looked at your materials and methods, because this is, of course, the disease here is haploinsufficiency, so that's just a little less. How much USP7 do you use in these assays?
Yeah, so we did a set of pilot experiments, and in our previous paper, it was focused more on degradation than on these non-proteolytic substrates.
Yeah, I should look back at that.
And we showed USP7 was kind of unique. We didn't test as many dubs as we tested in this paper, but USP7 was amongst them. And USP7 was really kind of unique in its ability to antagonize degradation of substrates in the Xenopus system. It was really good at antagonizing degradation. And we saw that in a dose-dependent fashion. USP7 is present, I think it's about 180 nanomolar in Xenopus extracts.
Oh, that's relatively low.
Yeah, we tested it in our screen, we tested it at 800 nanomolar. Because in our pilot experiments, we saw a nice level of ubiquitination. It's one of the first enzymes we tested this way. And we thought, okay, well, it's working at 800. Let's test everything else at 800. So, you know, that's the feature we kept in common across the screen. But we did do a dose response of USP7. I'm not sure. I don't think we put that data in the paper, but we do have that data. And you know, if that would be happy to share that with folks if they're interested in, you know, substrates that are, you know, particularly sensitive to USP7 in the system. But there's no doubt we got a dose response. If you test lower concentrations of USP7, you find fewer proteins for sure. But we haven't really looked at that set to say, oh, these are the ones that are super sensitive and these are the ones that are a bit less sensitive. I totally agree with you.
But my expectation sort of in the cell, just because there is a lot of it, but it seems important how much. So that equilibrium, is it available? Is it there? Must be important for when it acts.
I think that's true. And I'll say the other feature of our system that's different in human cells is there are no nuclei present in these extracts. Nuclear proteins are present, right? So the Xenopus embryo stockpile proteins.
Yeah.
So most of the proteins are there. You know, but the nuclear envelope itself, you know, itself is not. So you're not, you know, that has two effects. You know, we may lose certain proteins. We may not see them because they require USP7 to be in a nuclear compartment. We may find proteins that USP7 would normally not access because it's constrained in the nucleus. So I think that's an aspect of the work to sort of, you know, keep in mind when it's interpreted. That being said, as I mentioned, we find a lot of known USP7 substrates. So I think USP7 is able to identify or interact with a lot of these proteins independent of it being in the nucleus.
Yeah. Well, normally it's mostly in the nucleus, I guess. Thank you.
Thanks for your comments. Anything else? I don't know, maybe Amber's off the call here.
I think
she might have stepped out.
Becky, you're muted if you wanted to say anything.
Yeah, I was going to say, I didn't hear anything from her if she had to step away, but the presentation was great. Thank you so much. We definitely appreciate it for our organization. Like I said, when I spoke to y'all a little bit about Ella, we really, really appreciate everything that you all do.
Yeah, definitely my pleasure. Nice to have this chance to share the work with you all.
We'll say if we're done, we can go ahead and finish up then.
Okay.
All right, thank
Thanks.
you.
Nice to see you. Take care.
Bye.